Welcome everybody to this month's ERCC webinar. I'm Roger Alexander, the Scientific Outreach Coordinator for the Extracellular RNA Research Consortium. Really looking forward to today's speaker. She's Alissa Weaver, the Director of the Center for Extracellular Vesicle Research at Vanderbilt. She's a longtime member of the ERCC and has been doing important work understanding the mechanisms of our xRNA biogenesis and biology. And today she's going to tell us about new findings about the mechanism of biogenesis of EVs containing xRNAs at membrane contact sites of the endoplasmic reticulum. Please feel free to put questions in chat. We'll ask those first at the end of her talk and then have an open Q&A. Please keep yourself muted until then. And Alissa, we're looking forward to hearing from you.
Great. So as Roger mentioned, I was part of the first phase of ERCC, the biology, and we've been interested this whole time in the cell biology of how RNA-containing EVs are made. And as you all know, extracellular RNA is released from cells in many forms. It can go via extracellular vesicles, which will be my focus today, either coming from endosomes as exosomes or small EVs and budding from the plasma membrane as microvesicles. RNA can also be released from cells in non-vesicular particles. And Casey Vickers at Vanderbilt showed a number of years ago also that RNAs can bind to and be transported by high-density lipoprotein. So there's really a complexity of how RNA is released from cells. We've really been focusing on extracellular vesicles, in part because it is a way for the RNA to be transported in a protected form to recipient cells. So if you think about it, you know, the RNA is protected from RNase on the inside of extracellular vesicles. And then once the extracellular vesicles fuse with a target cell, it can access the cytoplasm to regulate gene expression and phenotype in the recipient cells. It's a bit harder to see how non-vesicular particles or other non-encased forms could do that. So even though this is an old cartoon, I think it's a very nice one because it sort of shows the different mechanisms that we broadly think about for exosome and extracellular vesicle biogenesis, and really places where RNAs are in this process. So the classic way that we think about that exosomes are formed within endosomes or otherwise known as multivesicular bodies is through the escort machinery. And so the escort machinery is recruited to ubiquitinated proteins, typically transmembrane receptors such as epidermal growth factor receptor. And then once recruited, the later components of the escort machinery will induce membrane curvature and budding of the vesicle to form an interluminal vesicle. So this is really the best understood and most studied mechanism to make an exosome. Other mechanisms that have been described involve formation of ceramide, which is a lipid that has a cone shape and can induce membrane curvature. And also tetraspanins, which are highly enriched in small extracellular vesicles, form these membrane-induced membrane domains that are like lipid rafts, but a little bit different, and have been shown to promote formation of extracellular vesicles as well, although it's not really understood exactly how that happens. Now, for RNAs and other cytoplasmic cargoes, how they get into vesicles and induce the membrane curvature and formation of the vesicle has been unclear. Ceramide formation has been implicated in it, but it's really not clear how the RNA gets recruited to a multivesicular body or to the plasma membrane to be selectively put into a vesicle. And we know that this is a selective process based on RNA sequencing, RNA binding protein studies. What we do know is that the RNA incorporation involves binding to RNA binding proteins. In this case, HNR and PA2B1 is shown to bind to microRNAs and be important for transport into an interluminal vesicle. So we became interested in this problem based on proteomic studies from our collaborator, Bob Coffey at Vanderbilt. And in fact, this was a focus of our phase 1 ERCC project, trying to understand how KRAS signaling affects RNA binding protein and RNA incorporation into extracellular vesicles. So what Bob's group had found was that the proteomes of small extracellular vesicles derived from an isogenic pair of cell lines, one of which was wild type for KRAS, the oncogene KRAS, or mutant for KRAS, which is an important oncogene in colorectal cancer, that those exosomes had very different proteomes. And in particular, the wild type exosomes had a very large fraction of RNA binding proteins, about 30%. And this was greatly decreased in the mutant exosomes. So based on that, we wanted to understand the molecular mechanism by which this was happening. And we focused for this paper that I'll just summarize in this one figure on Argonaut-2. So there was a lot of interest in Argonaut-2 at the time because it's an essential component of the RNA silencing machinery. And it could be a way to traffic microRNAs into vesicles and then help mediate downregulation in recipient cells. And so one thing that we hypothesized might be involved was regulation via phosphorylation. There were some sites that were known to be regulated by signaling. KRAS is an important upstream signaling molecule, upstream of MEK, ERK, and AKT. And so we sort of investigated that as the molecular mechanism by which differences in Argonaut were being found in extracellular vesicles. So this Western blot that I'm outlining here is with a phosphospecific antibody against serine 387 for Argonaut-2. I know it's kind of tiny, but I hope you can see that there's a darker band in the mutant KRAS exosomes compared to the wild type exosomes. And this is decreased if we use either a chemical inhibitor to AKT or to MEK. And MEK had been previously shown to signal at endosomes. So we were quite interested in MEK as a downstream signaling mechanism for KRAS to downregulate Argonaut-2 and prevent it from going into exosomes. Sorry, there's some road noise outside. Okay, so we also did colocalization studies. I don't have time to show you the images for this because this is an old paper at this point. But basically, what we had found was that Argonaut colocalized with CD63, which is our favorite tetraspanin marker of exosomes. So Argonaut colocalized with CD63 in wild type cells, but not in mutant KRAS cells. And so you can see quantitation here. And if we re-expressed an M-cherry Argonaut-2 in the mutant KRAS cells, we found that a non-phosphorylatable Argonaut at that serine looked like wild type cells in that Argonaut was now localizing to multivesicular bodies. But a wild type M-cherry Argonaut or a phosphomimetic Argonaut did not localize there. And this was paralleled by Argonaut-2 content in exosomes. So it was up with the non-phosphorylatable Argonaut and down in wild type and even more so with the phosphomimetic. We also found that microRNA content of exosomes was increased in the non-phosphorylatable mutant expressing mutant KRAS cells. And if we also did an Argonaut knockdown in the wild type cells, we found that there was a decrease in RNA content as assessed by nanodrop, or if we did qPCR for some candidate microRNAs that we knew were selectively exported into small extracellular vesicles, knockdown of Argonaut affected 2 of those, which was LET7A and MIR100. So we published this study in 2016, and we wanted to understand how this was working and investigate more how RNA binding proteins and RNA were associating with multivesicular bodies to get in there. So we knew phosphorylation, at least for Argonaut, was somehow related, but there are a number of other RNA binding proteins. And one thing that piqued our interest is previous findings that Argonaut 2 assembles first with mRNA. So Veriska machinery assembles on rough endoplasmic reticulum. So mRNA associated with ribosomes can actually recruit Argonaut and the rest of Veriska machinery. And this is the first site where it gets assembled and then can be shuttled to other places. And other work, and so actually a postdoc that was recruited to my lab to work on this, Vanashika Barman, she's on the call, and you'll see this is all her work. She had done some of this work, and she and others in her lab had also found that multivesicular bodies are probable sites of risk recycling. So we got quite interested in the concept that risk components and RNA associated with the endoplasmic reticulum might be making their way into endosomes and exosomes. And it's not just Argonaut, but other RNA granules that are known to be associated with the endoplasmic reticulum. This is just another example of a RNA granule known as a tiger domain or a TIS11B granule. So, RNA granules are these membraneless bodies. However, they seem to be highly associated with the endoplasmic reticulum. This, in this case, is called a tiger domain, but also there are other RNA granules like stress granules and processing bodies that are also known to be associated with the ER. So this made us think about contact sites between the endoplasmic reticulum and multivesicular bodies, and that this could somehow bring the RNA binding protein complexes into close proximity with MVBs and then into extracellular vesicles. So what are ER membrane contact sites? So this electron micrograph on the left is from one of Gia Volz's papers. They've done a lot of work on ER membrane contact sites. Basically, the endoplasmic reticulum makes contacts with many organelles in the cell, and a lot of very important exchanges take place between those organelles and the endoplasmic reticulum. So prominent examples are the exchange of lipids and also calcium. And we thought that this could be a potential mechanism to connect RNA binding protein complexes that are associated with the ER to extracellular vesicles. So the green in this image here is the endoplasmic reticulum. The central circle here is a multivesicular body, and you can see these very close contacts. So the membranes are not fused, but they are linked together by linker proteins. So as I mentioned, Vanashika Barman, who we all call BB, so I'm going to say BB from now on, really drove this entire project. And she began by looking to see if she could identify ER membrane contact sites with a proximity ligation assay. So what she did, or what this is basically you take primary antibodies against two different proteins that should be, that you want to see whether they're in close proximity or not, so less than 40 nanometers. And then there are secondary antibodies that have PLA probes. So there's a ligation and an amplification and detection. And basically, if two antibodies are in close proximity, you get a fluorescent dot. So if you look on the right, you can see red fluorescent dots. And she used antibodies against an ER marker, KDEL, and CD63, which I mentioned is our favorite multivesicular body marker. So on the right, the endoplasmic reticulum in the cell is labeled with a green fluorescent protein Sec61b fusion. And then the proximity ligation is identifying ER membrane contact sites with the red dots. So I hope you can see that these red dots are indeed localized along the endoplasmic reticulum. And these line scans, you can see the overlap of red and green. Okay, so BB next looked to see whether microRNAs are present at these endoplasmic reticulum MVB contact sites using the proximity ligation assay. And she found that indeed, she found examples of that where they were either completely overlapped or adjacent to each other. So again, on the right, you see a line scan where they're adjacent and then also overlap of the green and red signals for miR100, which is one of the microRNAs that we know gets exported into exosomes. LED7A is another one of those. And indeed, we find overlap and adjacency of those signals. She also did some, together with EM Core at Vanderbilt, electron microscopy to quantitate ER membrane contact sites. And we took some of those images, and Evan Kristofiak in the core, who's director of the core and extremely talented, helped us get some tomograms, and we could actually see interluminal vesicles being formed within multivesicular bodies at sites of contact with the endoplasmic reticulum. So the purple is ER, the blue is a newly forming exosome or interluminal vesicle, and the green is a multivesicular body. So given this suggestive data, we wanted to see if we could disrupt ER membrane contact sites and see what is the impact on RNA trafficking into extracellular vesicles. And so to do that, BB chose to manipulate a protein called VAP-A. So VAP-A is a membrane tether, so it's intrinsically linked into the ER, and then it binds to receptors on various organelles into the cell. There are a number of these tethers. VAP-A is one that's been very well studied, and it is particularly known to be important for formation of membrane contact sites with the plasma membrane and with endosomes, which are the two sites for EV biogenesis. And so she knocked down VAP-A with shRNA, making stable cell lines, and just checked whether this indeed affects contact site formation between the ER and endosomes. So on the left is, again, a proximity ligation assay. You can see lots of red dots in the scrambled control cells and fewer dots in the two different VAP-A knockdown cells. We also quantitated ER membrane contact site formation by electron microscopy. So again, scrambled is the wild type cells, and the endoplasmic reticulum makes close contacts with the multivesicular body. And this happens less in the knockdown cells. So you can see an example here where the endoplasmic reticulum is far from the multivesicular body. So we can measure the distance between the ER and the MVBs, and that's farther in the knockdown cells. And then if we count the contacts, if there are fewer actual contacts in the knockdowns compared to the scrambled. Oh, and I should mention, I don't have it listed everywhere, but this work that I'm telling you about today was recently published in Developmental Cell, so you can go check it out. So then she looked to see whether VAP-A regulated the number and RNA content of extracellular vesicles. And so what's shown in these graphs in the upper right is data from nanoparticle tracking, where we estimate the number of vesicles that are released from cells over the time that we collect our conditioned media. So we typically collect for 48 hours. And then we count the number of cells that are in the dish at the time that we collect the conditioned media. We do a cushion density gradient purification, which gives us very pure vesicles. And then we count the vesicles by nanoparticle tracking analysis. And so you can see that there is a small but significant effect of VAP-A knockdown on the number of both small and large EVs. And if we also looked at the size of the EVs by electron microscopy, this is negative stained preparations of our vesicles, there was a significant decrease in the size of the EVs coming from knockdown cells. We also wanted to look to see whether we were actually affecting multivesicular body biogenesis. And to do that, and to also look at the cargoes that are within, we leveraged an assay that's been used a fair amount by the field to look at luminal filling of multivesicular bodies. So this involves using a green fluorescent protein tagged version of Rab5. So Rab5Q79L is a dominant active form of Rab5. And it basically sort of traps the endosomes at the stage in between a Rab5 early endosome and a Rab7 late endosome. So it's sort of arresting it at the stage where the multivesicular bodies are forming. And so what happens is you get these very enlarged endosomes that are filled with interluminal vesicles and exosome cargoes, and it allows you to quantitate the luminal filling with different cargoes. And so this is just basically our positive control cargo CD63. It labels the endosomes very well. You can see the blue inside of the green is easy to quantitate. And we found that there was a decrease in both the size of those endosomes in the knockdown and also in the luminal filling of those endosomes. Now, there was a little bit of an increase in the number because there are, here's this the knockdown. There are more small endosomes in the knockdown, which we think is significant, suggestive anyway that there's less maturation of the endosomes into multivesicular bodies. And I'll come back to that assay because I'm going to show you luminal filling with RNA binding proteins and RNAs. But first, another characterization that BB did was to quantitate the total RNA per extracellular vesicle and also to carry out small RNA sequencing of the extracellular vesicles. So by A260, and we've found this also for another preparation of EVs with a qubit assay, we see a decrease in the total RNA per EV for both small and large EVs. And then from the RNA sequencing, we found that by principal component analysis, the microRNA signature of the knockdowns was very distinct from the scrambled control, both for small EVs and large EVs. And then if we looked for differentially secreted microRNAs, there were a number of them that were different. I should mention we also found differences in snow RNAs and tRNA fragments. I won't show you those, but they're in our paper. And And this is just a heat map of the ones that were conserved between small and large EVs. And some were predicted to be down and some were predicted to be up. In this case, what I'm showing you in D is a validation for microRNAs for the effect in VAP-A knockdown, small EVs, large EVs, and in cells. So BB carried out qRT-PCR. A couple of these, mirror 371, 372, were predicted from the RNA sequencing to be decreased in both small EVs and large EVs, and they are. Some of these other ones didn't quite make our statistical significance cutoff from the RNA sequencing, but we knew that they're ones that tend to be trafficked out into exosomes in those cells, and they were also down. They were down in our RNA sequencing, but just not statistically significant, but they are when you actually do the qPCR. And then interestingly, there was either no difference in the levels of those same RNAs in the cell, or in some cases, there was an increase in the levels in the cell. And we think this is suggestive that these are real sorting events. And actually, likewise, if we overexpress VAP-A, we see the converse of what I just showed you with VAP-A knockdown. So we see an increase in the number of small EVs and large EVs with VAP-A overexpression. So this is our nanoparticle traces. You can see the number and size distribution. And then if we quantitate the estimated secretion rate, we also see a significant increase in both the small and large EV secretion from VAP-A overexpressing cells. Likewise, there's an increase in the RNA per EV that we calculate from our nanoparticle tracking estimates of the number of extracellular vesicles and then the RNA that we quantitate. And when BB did qPCR for those same microRNAs that I just showed you, they're all increased in the VAP-A small and large EVs. In this case, they're decreased in the cell, again, suggesting that this is a sorting event that's actually regulating the levels of microRNAs in the cell. So what RNA binding proteins? I told you that RNAs rely on RNA binding proteins to be transported across, and we're thinking about these as complexes that get recruited to the endosomes to be put into a vesicle. And so BB carried out a Western blot analysis for known RNA binding proteins that have been implicated in carrying microRNAs into small extracellular vesicles. So I told you we had previously looked at Argonaut-2, and this is decreased in the small and large EVs that we purify from our knockdown cells. Likewise, HNRMP A2B1 and SYNCRIP are both down in the small EVs. HNRMP A2B1 is down in the large EVs, and we don't detect SYNCRIP at all in the large EVs. There was either no change in the levels of those RNA binding proteins in the cell, or for Argonaut-2, there was an increase in the cell. And that's all quantitated there. So basically, VAP-A is not only regulating transport of small RNAs, but also RNA binding proteins. Now, one thing that we like to do is to do some control experiments to see whether the RNA binding proteins and RNAs are on the inside or the outside of the vesicles. If it's a true packaging event, they should be on the inside of the vesicles. And so for proteins, we've typically used a dot blot assay in the laboratory where we dot various amounts of extracellular vesicles onto a nitrocellulose membrane. And then we probe those vesicles with antibodies, either in the absence or the presence of 0.1% tween 20. So a very mild detergent to just lightly permeabilize the extracellular vesicles and allow the antibody to enter. And so if we look at our small EVs in the absence of detergent, there's very little Argonaute 2 and no HNRNP-A2B1 that we detect, but we do detect CD63 because we're using an antibody to an extracellular loop of CD63. But in the presence of detergent, now we detect Argonaute 2 and HNRNP-A2B1 along with CD63. Likewise, for large EVs, we only detect Argonaute and HNRNP-A2B1 in the presence of detergent and not in the absence of detergent. And flotilin 1 should be on the inside. We see a little bit of it in the absence of detergent, but mostly it's on the inside of the large EVs. And then likewise, we use a different detergent assay to look to see whether the RNAs are on the inside or the outside. So in this case, we treat our purified extracellular vesicles either with nothing or with RNase A in the absence of detergent, or with RNase A in the presence of 1% Triton X100 as our detergent. And so basically what we see is that the RNAs, again, this is qPCR for various microRNAs along with U6, which is another RNA that is in vesicles. And we basically see that the RNAs are almost fully protected by the extracellular vesicles, and we only see the RNA disappear in the presence of detergent. And that's true for both small and large EVs. There's in some cases a little bit of a drop for LET7A and here for the large EVs, but the majority is protected. Okay, so as you know, extracellular vesicles can come from many sources, and when you purify them from conditioned media, you don't exactly know the source. So we did want to go back and look at endosomes and interluminal vesicle formation, and whether these RNA binding proteins and RNAs could be seen within those endosomes, and whether this was affected by VAP-A. And so we used, again, this GFP Rab5Q79L assay and looked to see whether a dye labeled MIR100, or I'm not showing you LET7A, could be detected inside of those endosomes. And indeed they are for our scrambled endosomes, but little to none for the knockdown endosomes. And that's quantitated on the right. And then for Argonaute 2, and also we did Syncrypt, but I'm not showing it to you here. Again, we see that there's a punctate staining for Argonaute in many different endosomes. And this is lost essentially for our VAP-A knockdown cells, again, quantitated. So we've quantitated this either as the percent filling of the endosomes, or the percent that we detect in the endosomes, or the intensity within the endosomes. Okay, so these data suggested to us that VAP-A was probably controlling a specific subset of extracellular vesicles. So I showed you that VAP-A affects very slightly the number of extracellular vesicles, but has more of an impact on the RNA content. So we wanted to see if we could subfractionate out some different vesicle populations and use that to try and understand the specific subpopulation that we thought VAP-A might be controlling. And to do that, we decided to test out a method that Clotilde Thierry's laboratory had published in 2016, which involves first making a small EV pellet and then doing a very fast flotation spin at 350,000 G for an hour. And this leads to essentially accumulation of the extracellular vesicles at these interfaces of the step gradient into a fraction 3 and a fraction 5, which they called light and dense fractions, and we also continue to call light and dense fractions. And they had found different cargos in there. And based on our looking at their proteomic data, we suspected that fraction 5 might contain some RNA binding proteins. So we went ahead and tried that, we being BB, of course. And so this is a Western blot of one of these density gradients. Here's as a positive control, cell lysates loaded on the right. And I hope what you can appreciate is that indeed we got the two different fractions. We found quite different cargos in fraction 5 when we looked for some of our favorite proteins that we thought were implicated in this biogenesis process. So actually VAP-A is present in fraction 5, as is argonaut-2, HNR, and PA2B1. Flotilin-1, which is an EV marker, is actually enriched in fraction 5 compared to fraction 3. And we also looked for LC3 because Jay Debnav's lab had published a recent paper showing that LC3 could induce formation of exosomes that had some RNA binding proteins and SNO RNAs in there. So we thought we would look to see whether our mechanisms were convergent there. Now, by contrast, in fraction 3, we saw more of some other EV markers, TSG101 and ALEX, which are, TSG101 is an escort machinery component. ALEX is a escort accessory that works together with syntonin. CD63 is enriched in fraction 3 compared to fraction 5. However, I will mention that these blots were loaded based on volume. And in fact, there's about a 10-fold enrichment that we found in terms of the number of EVs that went into this light fraction 3 fraction, as opposed to the dense fraction 5 fraction. So even though there are many fewer EVs in fraction 5, they are enriched for these RNA binding proteins and other proteins there. The other thing BB did, and this is true also for a second cell type that we looked at, HT1080 fibrosarcoma cells. And similarly, RNA binding proteins, LC3 and VAP-A are in fraction 5. And these other EV markers are more enriched, although also present, more enriched in fraction 3, although also present in fraction 5. And when BB looked to see, well, what about RNA itself, RNA enrichment? So, extracting the total amount of RNA and then dividing it by the number of EVs calculated from nanoparticle tracking, she found that the dense EVs were about 10-fold enriched for RNA, as assessed by A260. So fewer EVs, but more enriched in RNA. Okay, so to look at this a bit further, she also looked to see whether VAP-A was affecting one of these vesicle populations more than another. And so she looks at just EVs secreted from the cells. There's no effect of VAP-A knockdown on the light EV fraction, but there is a small effect on the number of dense EVs with VAP-A knockdown. And also there's an effect on the total RNA that we estimate per EV in the dense EVs. So decreased with VAP-A knockdown, but no difference in the light EVs. And when she did also a qPCR analysis for those specific microRNAs that we've been using to follow microRNA content of the extracellular vesicles, she found that the levels of those RNAs were all increased in the dense control EVs compared to the light control EVs, and that was also decreased with VAP-A knockdown. So the red is the dense EVs, and the gray and black are the light EVs, and then the darker red is the knockdown. So you can see there's really a selective effect of VAP-A knockdown on the dense EV population and no effect on this light EV population. And if we do our detergent sensitivity assay, we find that the RNAs are all on the inside of the extracellular vesicles for both light and dense EVs, because it only goes away with RNAs if we include the Triton X100 detergent there. Okay, what about RNA transfer to recipient cells? So I mentioned that extracellular vesicles are a good way to protect RNA and deliver it to recipient cells. And to look at that, we leveraged an assay developed by Jim Patton's laboratory at Vanderbilt, where they constructed luciferase reporter vectors, either a control vector or one which contains 3 copies of a perfect miR100 binding site into the 3' UTR of the luciferase. And you can then look at transfer of RNA from donor cells or extracellular vesicles to recipient cells and monitor downregulation of the luciferase in the miR100 reporter containing cells. And so panel B here shows a co-culture where we actually plate donor cells in a transwell insert above and co-culture them with recipient cells, which are in wells below. And I hope you can appreciate that there's no effect of any donor cell conditions for the control vector. And the conditions here are either a DKS8 donor or a DKL1 donor. Either an anti-miR 100 transfected into the donor cell or a control for that. But for the luciferase miR 100, we see a strong effect of the DKL1 donors to the DKS8 cell recipient cells. This is not affected by a control antagomer, but it's reversed with an anti-miR 100 antagomer in the donor cell. So indicating that the miR 100 is coming from the donor cells. If we now look at VAP-A knockdown, we see that the control donors downregulate the luciferase in the recipient cells, and this is reversed with VAP-A knockdown donor cells. And as I mentioned, there's more miR 100 in the DKL1 donor cells than the DKS8, which is why we see the difference. And there's less in the knockdown EVs. So if we just take the purified extracellular vesicles and we add them to the recipient cells instead of carrying out a co-culture, we see similar effects. So the control DKL1 donor downregulates luciferase in the DKS8 recipient cells, and this is reversed with VAP-A knockdown in the donor cells. If we look at using, instead of a cushion density gradient purified extracellular vesicle preparation, if we do our light dense EVs and we look to see whether light or dense EVs can transfer miR 100 to recipient cells, we find there's no effect of the light EVs. However, we do see an effect of the dense EVs. So again, the control DKL1 dense EVs downregulate luciferase in recipient cells, and this is reversed, having VAP-A knockdown in the donor cells. Okay, so how is VAP-A affecting RNA transfer and extracellular vesicle biogenesis into the EVs? So one possibility is that just not having these contact sites anymore is sort of removing the proximity of the RNA from the endosome. However, there are some other possibilities as well, because VAP-A binds to lipid transporters in other organelles, including cholesterol and ceramide transporters. And so we actually carried out an untargeted lipidomics analysis to see if there was a difference in the lipid content of small extracellular vesicles coming, well, and large extracellular vesicles. We looked at both of those coming from VAP-A knockdown cells. And we found many differences, but a very interesting one was differences in ceramides. So as I mentioned, ceramide is a lipid that induces membrane curvature and has been implicated in inducing exosome biogenesis. And this heat map that's shown on the left is showing all the different sphingolipids that were different between VAP-A knockdown and control EVs normalized to the cell lipid content of those same lipids, so that we're looking just at a sorting event. And you're looking at lipids that were altered in both the large and small extracellular vesicles. And so you can see the green is showing decreases in ceramides, and there were many, and sphingomyelins and other sphingolipids. But you can appreciate, I think, that there are many different ceramides that are downregulated in both small and large EVs from VAP-A knockdown cells. And so to validate that, we carried out another lipid mass spec experiment in which ceramide standards were used to quantitate the levels of ceramide, of those specific ceramides in the extracellular vesicles. And so we tested that out for several different ceramides. And what you can appreciate here is that VAP-A knockdown leads to a great decrease in these ceramides in both small and large extracellular vesicles compared to control, but there's either no change in the cell or it's not detectable in the cell. So this says submitted, but it's published, as I mentioned before, in Developmental Cell. So one of VAP-A's partners, lipid transport partners, is called ceramide transfer protein, or CERT. And so we thought that this might be a good possibility as a binding partner of VAP-A that could be mediating ceramide transfer from the endoplasmic reticulum into endosomes and helping to induce formation of the interluminal vesicles. So we carried out a immunostaining experiment to localize CERT to our Rab5Q79L endosomes. And I think what you can hopefully appreciate here, the red is CERT, the blue is CD63, which is our exosome marker, and the green is the limiting membrane of the multivesicular body, that we see CERT actually inside MVB on interluminal vesicles. And this is greatly decreased with VAP-A knockdown. There's little to no CERT on the inside of those endosomes. And that's quantitated underneath the images here. And so we actually knocked down CERT in the same cells, our wild type DKSA colon cancer cells, and found that they had a very similar phenotype to our VAP-A knockdown cells in that there was a small but significant decrease in the number of both small and large EVs, and that the same microRNAs were decreased in both small and large EVs, and either increased or no change in the cells from CERT knockdown comparison to control. Okay, so this was interesting because, the endoplasmic reticulum makes ceramide. It can be transferred to other organelles via CERT, although CERT has really mostly been studied as a way to transfer ceramide to the Golgi from the ER, not to endosomes, although there is one paper previously showing that it can do that. But really the field has focused on a different mechanism for generating ceramide to induce extracellular vesicle biogenesis. And this involves a protein called neutral sphingomyelinase 2, which enzymatically converts sphingomyelin to ceramide, thus generating this asymmetric shaped lipid ceramide. And so we wanted to see where neutral-sphingomyelinase was. The model is, or has always been, that neutral-sphingomyelinase 2 is, present on the endosome. It gets directly activated there, and this induces interluminal vesicle formation, exosome biogenesis. However, if that was what was going on in our system, then you wouldn't really need SIRT, ceramide transfer protein, because the ceramide could be just generated on site rather than transferred from somewhere else. So what we did was we immunostained for neutral-sphingomyelinase 2. What you're looking at here is the green is now a stain for endoplasmic reticulum. It's actually GFP-VAP-A. We use an M-cherry Rab5Q79L, which is pseudocolored white here, and neutral-sphingomyelinase 2 antibody, which is pseudocolored red here. And what we found was very interesting. These are two different examples. Actually, it's two different Z slices of the same cell I'm showing you, but we looked at a number of cells. And you can see that the red neutral-sphingomyelinase is closely associated with the green VAP-A labeled endoplasmic reticulum. And if you look at this line scan, you can see really the overlap is with the ER of the neutral-sphingomyelinase. Not so much, although they are adjacent, with the Rab5Q79L. Down below here is another example. This line scan is showing that although everything is in very close proximity and probably touching, the neutral-sphingomyelinase and the VAP-A are better overlapped with each other, and then probably touching the endoplasmic reticulum. We've also seen that VAP-A can really form these bridges to these neutral-sphingomyelinase positive structures. And I'm going to show you a movie of that. This is confocal microscopy taken at very high sampling and deconvolved, and then a 3D reconstruction that's formed. So this is just showing the bridge, VAP-A, really bridging this neutral-sphingomyelinase 2 structures to the endoplasmic reticulum. You can occasionally see a little faint hint of red there, but mostly it's the green that's touching the endosomes, not the red. This is now converting to a 3D reconstruction. You can see how, well, there's a VAP-A that's protruding into the endosome, a bridge to the ER. This is now moving through the slice. You can see the VAP-A bridging to the endosome. There is some neutral-sphingomyelinase that's there as well. It's also touching the ER. And lots of the neutral-sphingomyelinase is not connecting with the endosome except through VAP-A. So we have a lot more to do here. We think this is very intriguing. We want to try and figure out how these things are all connecting, whether neutral-sphingomyelinase could be really bridged, as these data suggest, through VAP-A to endosomes. And here is our model. So we think that endoplasmic reticulum membrane contact sites are a very important place for biogenesis of RNA-containing extracellular vesicles. So VAP-A would link to ceramide transporter on the endosome and transfer of ceramide, either that which is synthesized as it's known to be in the endoplasmic reticulum, or made by neutral-sphingomyelinase in some kind of a ER-associated organelle, could be transported by SIRT to induce membrane curvature and formation of a vesicle. And the association of RNA binding protein and RNA granules with the endoplasmic reticulum is, we think, probably the source for the RNA that's coming in. So these events must be coordinated, although we don't totally understand how at this point. This work was virtually all done by Banashika Barman, who is now an instructor in my laboratory and will be on the job market very soon for faculty positions. She had some wonderful imaging help from Bongwon Sung in my laboratory. He's a research associate professor and a fabulous microscopist. We had a lot of help from our collaborators. This work really started in the sort of last year of our ERCC phase 1 project and is now continued as part of a program project grant that we have together. And thank you very much. I think I'll stop there, take any questions.
So Alissa, we have a lot of questions. I don't know if you have a couple minutes after the hour to take them, or...
Sure, yeah, absolutely. Yeah, so I can just start with some of these from Alex. So we believe that the RNA binding proteins do accompany the extracellular RNA into the extracellular vesicles. We are gearing up. I have a new student that will be working on some cross-linking approaches to be able to identify how these complexes are being made and potentially altered throughout the life cycle of, putting them into an exosome, and then what happens later in recipient cells. There's a question about Argonaut 2. So we are actually working on another, so Argonaut has been very controversial because many people have found it to be a contaminant, and indeed a lot of Argonaut 2, especially in serum, which is a major media component, is non-vesicular. As our work also shows, Argonaut transport into EVs can be downregulated by growth factor signaling. So we are currently in sort of late stages of working on a paper to show that, in fact, the conditions under which you isolate your extracellular vesicles, serum or no serum, and growth factors will greatly affect whether you're detecting extravesicular Argonaut or intravesicular Argonaut. So our opinion is that it's a real component, but it is greatly masked if you, for example, have serum, or if you have a lot of dead cells that would be releasing the non-vesicular complexes into the media. Of course, Argonaut is not the only important RNA binding protein for transport of RNAs into extracellular vesicles. And, as I showed you, we were also tracking HNRP, A2B1, and SYNCRIP, and would like to expand that. Okay, so let's see. There's a question about RNA equivalent of the dot blot assay. Yes, we used. Do we target specific RNAs? Yes. So we tracked microRNAs with qPCR in the presence and absence of Triton X100 and RNase, and found that the majority of the RNAs we were looking at were protected inside of the extracellular vesicles. We did not use proteinase and then RNase. I've been a little bit leery about that because I've heard that it can disrupt vesicles on their own, but we just really haven't tried using proteinase K in these assays, so we don't really know. Okay, there's a question about which microRNA we've taken as a housekeeping control. So we actually use U6, which is a SNO RNA, or it's a small nuclear RNA, and not a microRNA. So that's a common one that people use to normalize it. I don't know why, but it seems to be transported selectively into extracellular vesicles, but it's often used as a housekeeping control because like when we were doing our Argonaut work, we found that the Argonaut was not affecting the U6, but it was affecting the microRNAs. So U6 was a good control for those in that cases, but it generally is present at pretty high levels in the exosomes. So there's a question about how do we know that dense EVs are actually EVs and not non-vesicular particles? I guess we don't know absolutely that there aren't some non-vesicular particles in there, but they are floated from the bottom of the density gradient, which is a characteristic of vesicles and not non-vesicular particles. And then we also carried out our detergent sensitivity for the RNA with the dense EVs and found that the RNA was only degraded if we had Triton X100 there. So although we can't rule out that there aren't some non-vesicular particles in that fraction, the RNA part of it is inside of EVs that we were assessing.
Alissa, Alex and I had a related question. So you had small and large EVs that you differentiated by CD63 and flotilin surface markers. And then with the Terry subfractionation, you have the light and dense subpopulations. Mm-hmm. And Alex mentioned a couple of known subpopulations of EVs. Do you understand the connection between those things? Do you know like which ones are the same or?
I guess I don't totally understand your question.
So like, for example, is the CD63 versus flotilin small and large classes, do those correlate with the light and dense fractions in the Terry assay at all?
Okay, so flotilin we typically find in both small and large EVs. CD63 we typically find in only small EVs. For the light dense, it was quite interesting because flotilin seemed to really be enriched in the dense EV population, whereas all the other markers were in both light and dense. So CD63, Alex, TSG101, and syntenin were in both light and dense EVs. We never did like an equal loading of particles on a gel, so I can't tell you if they're totally different. I mean, if they're different or not on a per vesicle basis. All I can tell you is that we really didn't see much flotilin in the light EVs, despite having 10 times more extracellular vesicles in that fraction.
So that would argue that large EVs are enriched in the dense fraction?
Okay, so the light dense prep we only did with small EVs. So
okay.
Can't say anything about large EVs there.
My question was actually, how do these light and dense correspond to those light and dense found by blood pulse group in 2017 and found indirectly by us, a few years ago based on their cargo? Is there some correspondence between those classes?
Between what Clotilde found and we found?
Yeah, the light and dense EVs. They're found in cell cultured media, by Cecilia Laser from blood pulse group in 2017, I think. These two distinct classes, by density.
We haven't done proteomics on them yet. That's something that we would like to do. So we're trying to scale up in order to do, we're now using hollow fiber bioreactors so that we can scale up the number of extracellular vesicles and get some preps that have enough protein to do proteomics on the light and dense EVs. What we had noticed in Clotilde's proteomics data was that there were a number of ribosomal proteins in the dense EV fraction. And as I told you, ribosomes are closely associated with the endoplasmic reticulum, and those proteins are some of the most common RNA binding proteins to be found in extracellular vesicle preparations. So just based on that, we thought that the dense fraction would have our RNA binding proteins. We never looked for ribosomal proteins there, but we looked for the ones that, we track through our process, and that's what we found there. But I think that having, well, yeah, we can only say so much about that because we didn't do Western blots loading equal vesicle numbers, and we haven't done proteomics. So there's not
also RNA-seq. Have you tried RNA-seq? Or?
We have not done RNA sequencing. Again, we have to ramp up the amount of vesicles that we can get in order to do that. Yeah. Because
we have published these profiles, characteristic of dense and light ones, and comparison of our small RNA-seq profiles may tell us whether there's any correspondence.
Yeah, no, it would be very interesting to compare.
Mm-hmm. Thank you.
Let me ask, sorry for the confusion, let me ask a couple specific chat questions from Tutran Nakmin. Thanks for the great talk. Is there a preference of site for VAP-A to support EV formation, for instance, to CD63 rich regions? How about other EV tetraspanins such as CD9? Has there been an interactome analysis of VAP-A in real time during EV biogenesis? In KD of VAP-A cells, if the RBPs and associated RNAs are not in EVs, did you see them sorted to other organelles or cellular sites?
Well, we have not looked at tetraspanins other than CD63 because those have been found by Clotilde and some other groups to be on the plasma membrane, although some people have found them to be in endosomes. By contrast, we've done a lot of work characterizing CD63 in our lab and found that it's a really good exosome endosomal marker. We developed a live imaging reporter based on that, et cetera. So we were really using CD63 just as a really great positive control and endosome marker. One thing that was interesting was that, we didn't see like endosomes that had all the RNA or RNA binding proteins like concentrated in them and then nothing elsewhere. It really seemed like it was sort of punctate and sparse throughout many endosomes in the cell, suggesting that, lots of endosomes may be touching the endoplasmic reticulum, and they may have already made other exosomes with other cargoes along the way, and then they're also making these ones that depend on VAP-A and have the RNA binding proteins and have the RNAs in there.
That leads to a question Alex asked early on that I don't think you addressed. The RBPs that are interacting with RNAs, do you have information about whether the RBPs end up in the vesicles, or do they just sort of chaperone them there, get the RNAs into the vesicles and stay in the cell, or is it not generalizable?
So we know that the RNA binding proteins that we were tracking are inside the exosomes and inside interluminal vesicles. What we don't know is whether they are bound all the way to a specific RNA. So we just don't know that. I think that's a really, important question, but at this point we don't know.
I always hear of CD63, CD9, and CD81 sort of as the triumvirate of tetraspanins. You're saying that CD63, you have sort of more confidence that is a strong marker for vesicles.
I think it's a strong marker for exosomes that are derived from multivesicular bodies. CD9 and CD81 are great markers for small EVs, but work from other people have found that small EVs can be made at the plasma membrane. And at least in some cases, those are primarily localized to the plasma membrane and probably making very small microvesicles on the plasma membrane.
Santosh Yadav asks, the knockdown of VAP-A and other proteins only reduced the number and size of EVs, but still the EVs were synthesized, suggesting that other pathways are also available for EV formation. What is your view?
Yes, absolutely. I mean, I think this is a very small subpopulation of EVs that is carrying the RNA and RNA binding proteins, and that is subject to regulation by VAP-A. So that was part of why we did that dense and light, EV prep, because that really helped us show that a little bit better, that there's this, small vesicle population that we can separate out with the density gradient. That's only 10% of the vesicles that are being made, but it's carrying them, it's tenfold enriched in RNA compared to the other vesicles, and it's selectively controlled by VAP-A. So VAP-A doesn't seem to be touching most of the vesicles that are formed, just, these ones that are containing RNA and RNA binding proteins.
That relates to another of my questions, which is what led you to VAP-A specifically at that particular membrane contact site tether? If you did a sort of systematic siRNA knockdown of other known tethers, first of all, how many are known? And would that be a good way to sort of try and shine light on the other set of subpopulations of EVs that RNA?
Yeah, so there are other membrane contact linkers. They, at the time we started this project, they had not been as well studied. So VAP-A is one that had been really characterized to be important for forming these links with the plasma membrane and endosomes, but people keep finding new linkers over time. There's another VAP protein called VAP-B, which seems to be more important for connecting to the mitochondria. However, VAP-A and VAP-B can not only homodimerize, they can heterodimerize with each other. So that gets a little bit complicated. Some people have done double knockouts, but I think cells would start to get unhappy if we manipulated VAP-B with the mitochondrial connection.
Let's make this the final question from Srima Yukoner. Is the RNA population of exosomes, does it reflect the RNA content of the cell? I mean, if there's an upregulation of a particular small RNA in the cell, will the exosome have the same upregulation level, or the loading of small RNA is independent of its upregulation in the cell?
Well, there have been a number of studies that show that at least some RNAs are selectively sorted. So if you compare the exosomes and the cell levels, they're not, you don't have just, it doesn't completely reflect the levels in the cells. And in fact, that was one of the papers early on by that Jim Patton at Vanderbilt did with these this whole colon cancer model system that I was showing. And they found that, some RNAs were selectively sorted based on KRAS signaling. Some were enriched in exosomes compared to cells. And then, some were preferentially enriched in cells compared to exosomes. I will say, when we analyze our RNA sequencing data, so we specifically normalized it to the cell level. So we were only looking for RNAs that were selectively sorted and that were different according to VAP-A after we normalized to the cell levels, just in case VAP-A was affecting any RNAs in the cell. We really wanted to look at specific sorting events.
I wonder if I could get your take as a final question on the ubiquitous, I always ask question about the disconnect between the number of RNAs, the RNA quantitation versus the EV quantitation, and its implications for functionality in target cells. One of your graphs showed tenfold enrichment in one population of EVs, but they were all like one RNA per thousands or tens of thousands or more vesicles. What's your take on that problem?
Yeah, I mean, so our quantitations were really based on nanograms per vesicles. So we haven't gone to like copies of RNAs. I couldn't tell you about specific copies of individual RNAs. It's probably something we should also do in the future. But I do think that being able to, pin it down to a subpopulation of vesicles like this, plus being able to show a specific biogenesis mechanism, I mean, I think it shows that, of course, it may depend on the cell type or the cell situation, but by and large, at least from what we were looking at, the vesicles that are carrying the RNA are a very small subpopulation of the total extracellular vesicle population that we're purifying from cells.
So that basically once people are, based on this work and other work, better able to subfractionate, they will find the subpopulation of vesicles with all the RNA in it and can then check the function of those in target cells.
That's what I think. I think that there's two things. One, it's a very small subpopulation, and that accounts for why people find so little RNA in extracellular vesicles. And I think it's also probably highly subject to regulation. So it could be that cells really care about whether they're exporting RNA or not. So, I mean, as you all know, extracellular vesicles are really heterogeneous, and they are, whether or not you're exporting a cargo is likely to be a very regulated effect. And another thing we found was that often the RNA and RNA binding protein levels were changing in the cell if we saw a difference in the content in the exosomes. So for example, when we saw RNAs go down in the exosomes, we often saw them go up in the cell, and vice versa. When we overexpressed VAP-A, we found levels of those same RNAs going up in the EVs and down in the cell, suggesting that there's not only a specific sorting event that's taking place, but it also begs the question as to whether this could be a way for cells to control the cellular levels of these things, not just whether it's going on and communicating with other cells outside.
We'll end it there. I could ask a half an hour more worth of questions, but I appreciate your accommodating us with the extra time. And as always, your work is fascinating, and look forward to seeing what the next phase holds.
Thank you so much. This has been great.